Hi everyone, Tanima at this end. You're watching this video in my YouTube handle at @justtanima_Barbie. Just to let you know, my YouTube venture is totally a personal endeavor as a voluntary virtual social outreach, and it has nothing to do with my profession where I'm engaged with research and teaching in the field of molecular biology and biotechnology in an institution in India. I personally believe that education should be freely accessible to all, and with this in mind, I have initiated a series on cancer biology. This is my 10th video on cancer biology. In continuation to my 9th video, the present video intends to discuss some more perspectives of microenvironmental mechanisms of epigenetic reprogramming in the category of non-mutational epigenetic reprogramming as another hallmark of cancer. The link of the 9th video is provided in the description box. The present video partially discusses the paper entitled Hallmarks of Cancer, New Dimensions. The DOI of the same has been pasted in the description box. I would also request you to turn on the subtitle or caption button on your screen in case you find difficulty with my deliberation. Do keep watching. Another compelling line of evidence supporting microenvironmentally mediated epigenetic regulation pertains to the invasive growth potential exhibited by cancer cells. A quintessential illustration of this phenomenon is the reversible enhancement of invasiveness in cancer cells located at the peripheries of numerous solid tumors, process governed by the developmental regulatory mechanism referred to as the epithelial to mesenchymal transition or EMT. Importantly, a principal regulator of the EMT, that is ZEB1, has been recently elucidated to promote the expression of a histone methyltransferase ACTD1B, which subsequently sustains ZEB1 expression through a positive feedback loop that pertains to the invasive EMT regulatory state. A prior investigation similarly documented that the induction of EMT via the upregulation of a related transcription factor SNAIL1 resulted in significant modifications to the chromatin landscape, consequent to the activation of several chromatin modifiers whose functionality was unequivocally essential for the preservation of phenotypic state. Moreover, a comprehensive array of conditions and variables to which cancer cells residing at the peripheries of tumors are subjected, encompassing hypoxic environments and cytokines released by stromal cells, can unequivocally trigger the epithelial to mesenchymal transition or EMT and consequently enhance invasiveness. A salient illustration of microenvironmental programming pertaining to invasiveness, ostensibly dissociated from the epithelial mesenchymal transition or EMT program, encompasses autocrine activation in pancreatic cancer cells and other cell types through interstitial pressure-induced fluid dynamics, facilitating a neuronal signaling pathway that entails the release of glutamate alongside its receptor, NMDAR. It is noteworthy that the prototypical rigidity of numerous solid tumors manifested through extensive modifications to the extracellular matrix or ECM that encase these cells exerts widespread influence on the invasive and various phenotypic attributes of cancer cells. In contrast to the normal tissue ECM from which neoplasms arise, the tumor ECM is generally defined by augmented cross-linking and density, enzymatic alterations and modified molecular composition, which collectively orchestrate partly via integrin receptors interacting with ECM motifs, stiffness-induced signaling cascades and gene expression networks that promote invasiveness along with other characteristic features. In addition to such regulatory mechanisms conferred by the physical tumor microenvironment, paracrine signaling involving soluble factors released into the extracellular milieu by the diverse cell types inhabiting solid tumors may also play a pivotal role in the activation of several morphologically distinct invasive growth programs, of which only one, designated as mesenchymal, appears to incorporate the previously mentioned EMT epigenetic regulatory mechanisms. So viewers, hope the video could provide some idea on the topic of microenvironmental mechanisms of epigenetic reprogramming. Please do not forget to like and share the video and also subscribe to my channel. Here I would like to mention that the references that were used for preparing the video have been pasted in the description box. If you're interested to look into the details, you might check the same. Hope to meet you soon with yet another video on cancer biology. Till then, do take care, keep up, and bye.